Hello and welcome everyone. Today we will discuss about nucleic acid. The lecture about nucleic acid will be covered in two lectures. So this is lecture one. In this lecture we will discuss about the basic introduction and structure of nucleic acid. And lecture two we will discuss about the detailed types of nucleic acid. So first of all, we will discuss about the nucleic acid introduction. Nucleic acids are complex macromolecules and they are organic in nature. So this is a class of organic molecules which are large, complex, and also known as macromolecule like carbohydrates, proteins, and lipids. Nucleic acid was discovered in 1869 by Friedrich Miescher, who isolated a compound from the nuclei of WBC white blood cells, which was taken from the pus cells of discarded surgical bandages. So the chemical nature of this compound was unknown at that time, and thus Miescher named that compound, which was obtained from the WBC cells, nuclein, because it was isolated from the nucleus or nuclei of the cell. So the structure of this compound was unknown at that time. Then Richard Altman in 1889 renamed nuclein to nucleic acid, which is now known as DNA. So the name nucleic acid was given by Richard Altman. Nucleic acid occurs in two forms, which is first one is RNA, also known as ribonucleic acid, and DNA, which is known as deoxyribonucleic acid. DNA is located inside the nucleus, while RNA is formed in the nucleus and then exported to cytoplasm to perform different types of functions. So nucleic acid plays an important role such as storage of genetic information, which is in the form of DNA, and transmission of genetic information in the form of RNA, and protein synthesis. In protein synthesis, this mRNA is converted into proteins, and the three types of RNA, messenger RNA, transfer RNA, and ribosomal RNA, all take part in this process. So these are some important roles which is played by the nucleic acid, especially DNA and RNA. The transfer of genetic information which is present in DNA occur with the help of replication, transcription, and translation. So these three are the basic processes in which the genetic information which is present in DNA transfer from generation to generation. So first is replication. In the replication process, the identical copy of DNA is made. So this process is very important because it preserves the genetic information, because that copy of DNA which is made in the process of replication is transferred into new daughter cells. Then the next one is transcription. Here in this process, the genetic information are transferred to mRNA from DNA. The last one is translation. Here the genetic messages are decoded to make proteins. So the genetic messages which are transferred from DNA to mRNA are decoded here, and that is used to make a specific protein. So in this figure you can see these processes are shown. This is DNA. DNA is in DNA process. This DNA replication occur and it's make a copy of itself. From DNA the information is transferred in the process of transcription to RNA, especially messenger RNA. Then from mRNA, the information are used to make a protein in a process known as translation. So these three are the basic processes in which the transfer of genetic information occur, which is present in DNA. Next is the structure of nucleic acid. Nucleic acid contain DNA and RNA, and we know that both DNA and RNA are polymers. Polymers mean large molecules which are made of organic molecules called nucleotides. So nucleotides acts as a monomer of nucleic acids. Nucleotides function as a metabolism energy in the form of ATP, as a cofactor in the form of NAD, and in signal transduction as a form of cyclic AMP. So these are the role, some of the important roles which is performed by the nucleotides. ATP, we already know about the ATP, it is the energy currency of the cells. Cofactors, these are the non-protein part of enzymes, and these are organic in nature. NAD, FAD, these are coenzymes. So in cofactor, further these are classified as coenzymes because these are organic in nature. And signal transduction, cyclic AMP, these are important types of nucleotides which perform very important function inside the cell. Typically, a nucleotides consist of three parts. First one is a nitrogenous base. Next is 5-carbon sugar, which is known as pentose sugar. And third one is a phosphate group. So these three entities are subunits combined to make a specific protein, a specific nucleotide. So these are the basic components of nucleotides. So you can see here in this figure, this is the sugar which we already discussed. It is a pentose sugar. You can see this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So it contains 5 carbon. This is carbon 1. At carbon 1, nitrogen is base is attached. This is adenine. And this is at carbon 5, phosphate group is attached. So this is a typical structure of a nucleotides. So now we will discuss each part of a nucleotide in detail. First one is nitrogenous bases. These are organic molecules made of nitrogen containing ring structure, and these are classified as a purine and pyrimidine. So first, purine is a double ring structure and it includes adenine and guanine. Pyrimidine is a single ring structure which includes thymine and cytosine and uracil. Uracil is present in RNA. And the pairing will be among adenine and thymine. Adenine will be paired with thymine and guanine will be paired with cytosine. In RNA, uracil is present which replaces thymine. So these are examples of nitrogenous bases which are organic molecules, and these are made of nitrogen containing rings. So you can see here, this is adenine. This is a double ring structure, and you can see here nitrogens are present. So this is the nitrogen ring structure. This is the numbering is also shown. This is carbon nitrogen 1. This is 2, 3, 4, 5, 6, 7, 8, 9. And this is guanine. You can see here, this is also a double ring structure, and it is an organic molecule which is a ring made of nitrogen containing compounds. So you can see here also the numbering is the same as adenine. These are pyrimidines. You can see here these are the single ring structure, and the numbering is shown here 1, 2, 3, 4, 5, 6. All are the same. You can see slight differences in the structure. So you can see here, this is the detail is given in this figure. So uracil, this third one, uracil, this is only present in RNA, while cytosine and thymine are present in DNA. In this table, the nucleotide and nucleic acid nomenclature is given. You can see here, this is bases, purine and pyrimidine, and this is nucleoside, and this is nucleotide. And in which type of nucleic acid it is present. So you can see here purine, adenine, it is also known as adenosine in RNA and deoxyribonucleic in DNA. So in the nucleotide, you can see it is known as adenylate in RNA and deoxyribonucleic in DNA. Guanine, you can see this is another purine, it is known as guanosine in RNA and deoxyguanosine in DNA, and it is in guanylate and deoxyguanylate in terms of nucleotides. So you can see the pyrimidines, when it is in the form of nucleoside, it is known as cytidine, and which is present in RNA, and deoxycytidine present in DNA. In the form of nucleotide, it is known as cytidylate, and which is present in RNA, and deoxycytidylate present in DNA. Thymine is known as thymidine or deoxythymidine, thymidylate or deoxythymidylate present in RNA and DNA. Uracil, which is only present in RNA, is known when it is a nucleoside is known as uridine. When it is a nucleotide, known as uridylate, which is present in RNA. So these are the nitrogenous bases and its nomenclature when it is a nucleoside and nucleotide. Nucleotide, we already discussed that it consists of nitrogenous base, sugar, and phosphate, while nucleoside lacks a phosphate group and contains nitrogenous base and sugar. Next part of nucleotide is sugars. DNA and RNA contains 5-carbon sugar, which is known as pentose sugar. It is called deoxyribose in DNA and ribose in RNA. Because the difference between this is known as deoxyribose and ribose, because it differentiates between at carbon number 2, because ribose contain OH group at carbon number 2 and deoxyribose has only hydrogen group attached at carbon 2. So this is, you can see here, this is deoxyribose. This is a pentose sugar. The carbon numbering is given, carbon 1, 2, 3, 4, 5. So at carbon 2, the main difference between ribose and deoxyribose is at carbon 2. You can see at carbon 2 only hydrogen is present, and in RNA or ribose at carbon 2 OH group is present. So it is known as ribose and it is known as deoxyribose. The carbon atoms of pentose sugar is numbered as prime 1, prime 2, prime 3, prime 4, and prime 5. Dash means prime, and it indicates that the carbon belongs to sugar molecules. So only the carbons which is present in the sugar molecules are labeled as prime. So to denote it that it belongs to sugar molecules. Sugar occupies a central position in nucleotides with nitrogenous base attached to carbon 1 and phosphate to 5 carbon of the sugar. So you can see here, this is a nucleotide. The sugar is present at the center. At carbon 1, nitrogenous base is attached. At carbon 5, phosphate group is attached. In pyrimidine, nitrogen at one position bonded to carbon 1 of the sugar, and in purine, nitrogen at 9 position is bonded to carbon 1 of the sugar. So you can see here, this is carbon 1 of the sugar, and this is a purine. So carbon nitrogen at 9 is attached here to carbon 1 of the sugar in a bond known as glycosidic bond. And when a pyrimidine is present, so here pyrimidine will bond with carbon 1 at nitrogen 1. So the nitrogen present at 1 will be bonded with carbon 1 in pyrimidine, while nitrogen at 9 will bonded at carbon 1 at the sugar and a bond known as glycosidic bond. And the 5 at 5, the phosphate will be bond here in a ester bond. So this is an important point. You have to know that in pyrimidine, nitrogen at 1 position is bonded to carbon 1 of sugar, and in purine, nitrogen at 9 position bonded to carbon 1 of the sugar. The last part of a nucleotide is a phosphate. Nucleotide contains a single phosphate group or a chain of up to 3 bonded to 5 carbon of one sugar and 3 of the next sugar. So this is the last part, the phosphate group. You can see here, it attached to the nucleotides. You can see at 5 of one nucleotides and to 3 carbon of the next nucleotide to form a nucleotide structure. A nucleotide lacking a phosphate group is known as nucleoside. So we discussed in the last slide that nucleotide contain nitrogen. This figure you can see here, this is a nucleotide. So it consists of phosphate, sugar, and nitrogenous base. And when phosphate is removed, it is known as nucleoside. So nucleoside only contains sugar and nitrogenous base. So next we will discuss how 3 to 5 phosphodiester bonds occurs in DNA and RNA. So in a nucleotide, phosphate group is attached to OH group at 5 carbon of sugar 1 of one nucleotide, and the OH group at carbon 3 of the sugar of the next nucleotide, forming a 3 to 5 phosphodiester bond. So we know and discussed earlier that phosphate attached to the OH group present at 5 carbon of one nucleotide, which is the sugar of one nucleotide, and then the OH group at carbon 3 of the next nucleotide sugar, forming a 3 to 5 phosphodiester bond. And also in DNA chain, all 3 to 5 OH group are involved in phosphodiester bonds except the first and the last nucleotides. So except first and last nucleotides, all 3 and OH groups will form phosphodiester bond. So why first and last nucleotide lacks phosphodiester bond? Because the first nucleotide has a phosphate not bonded to other nucleotide, and the last nucleotide has a free 3 OH. Thus each DNA contains a 5 to 3 free end. So you can see here in this figure, it is given the 3 to 5 phosphodiester bonds formed between nucleotides and DNA and RNA molecule. This is a molecule of DNA and this is a molecule of RNA. So you have to remember this. We discussed that the 5 end, the last 5 end and 3 end is free. So you can see this is the last 3 end. It contains an OH group. It is free. It is not bonded to any nucleotide. And this is the last, the 5 end. You can see the phosphate is free and can bond to any nucleotide, but it is free because this is the last end. So 5 and 3 end are free in DNA and also in RNA. So now this is the first nucleotide. It contains sugar, nitrogenous base, and phosphate. So phosphate is attached at carbon 5 of this sugar, you can see. And the next group, the phosphate is next attached to the nucleotides. The sugar of the next nucleotide at carbon 3, you can see here. This is carbon 5, this is carbon 3, and you can see here this is phosphodiester bond. This is shown here, this is a phosphodiester bond. So you can see this is the carbon 3 have phosphate, the carbon 5 also has, this is the next nucleotide. You can see this nucleotide, this is the carbon 5, it attaches to phosphate, and this is the another nucleotide, the carbon 3 also attached to phosphate. So it makes phosphodiester bond. So this is the phosphodiester bond. It is in DNA and also in RNA the same. So you can see here, this is the RNA, it has a free 5 or 3 end. This is the first nucleotide, the 5 group attached to phosphate, and the 3 also attached to phosphate. So this is the 3 to 5 phosphodiester bond formation, both in DNA and RNA. So this is all about the introduction and structure of nucleic acid. Thank you for watching.